New to this type of format, essentially what I do is normally I'll post a whole bunch of different topics for people to choose from, and then they pick what it is that I talk about every Saturday. So I was in London last week, and one of the people that I connected with is named Martin Jones. He works at the Francis Crick Institute. And you can follow him on Twitter at martinjones78. And he gave me a tour of the Francis Crick Institute in London. And he talked about his project associated called Etch. And so today, I'm going to tell you all about his program and how you can get involved in his research. So let's get started. So hello science lovers, today we're going to talk about Etch and how you can get involved in this particular research and why people are important with this citizen science project. Must have played Etch-a-Sketch as a kid, probably, and it's not unlike playing Etch-a-Sketch with this particular project. So here's me. I'm going to talk to you a bit about what the Francis Crick Institute is. It's located in London, and so there's a very happy scientist Mel right outside the Crick Institute. That was last Thursday, I believe. I was there, and it was a really, really awesome experience to be a part of that. So let me pop over here. So what I want to talk to you about today is the Francis Crick Institute, the work that they do. They're really heavily involved in several different types of projects, but the project I'm going to talk to you about today is called Etch, and it's a great program, especially if you're a science enthusiast or if you're a teacher and you have a classroom. You can get your kids involved or your teens involved or your students involved, and this is also good with university as well. So here is more of the Crick Institute. If you happen to be in London, you can go into the Crick Institute and walk into their little section. They have a section over here on the right. You're able to walk in and they have all of the information there about all the work that they do. And you can also leave questions for different scientists. So here is more of the inside of the Crick Institute. The thing that I really like about this setup, as I was going through, they have all of their office space and all of their labs are connected together. It's a gorgeous building on the inside. So all of these floors you see, there are offices attached to the labs so people can interact with one another from different divisions, and that's actually quite important in science to be able to talk to other people in order to bounce off your ideas. So the Crick Institute is heavily into scientific collaboration. So here is Martin Jones. This is the gentleman that actually took me around Francis Crick Institute and talked to me about his research. And there is the Citizen Science Etch website. So let's talk a bit about what their research is. Essentially, Etch, it's related to what a particular branch of cellular research involving imaging, 3D mapping. So they essentially are trying to map the nucleus on the outside, the nuclear envelope. And they do this through electron microscopy. So they scan various layers of the cell, and then they go back through and they highlight the nucleus, and then they piece it all together, and then you get a 3D image. So that's essentially what this type of research is. Right now they have 3,710 volunteers. These are people that they call citizen scientists that are working on this project. Now, you might think, okay, why is this research important? Why do we care? Why should I be involved in this? Well, here's the thing. The shape of a nucleus is linked to various types of diseases. So if we're able to compare the shape of a healthy nucleus along with disease nucleuses, we can get a better understanding. Yes, where can you sign up? You can actually Google Etch a Cell. If you just Google Etch a Cell, Francis Crick Institute, it'll pull up their Twitter, and it'll also pull up the direct site, and I'll show you where you can go to get involved. So the whole idea is once they determine the shapes of nuclei associated with various diseases, compare it to healthy nuclei, that can give us a database of all kinds of nuclear shapes associated with diseases, and it can also lead to better detection methods. Francis Crick Institute looks like science from iRobot. It's gorgeous. I feel so lucky to be able to tour such a fantastic facility. And Martin is a wonderful guy. Martin Jones 78 on Twitter, if you want to ask him directly any questions. He's the outreach person for their citizen scientist project. So this is why this research is important, and this is why your help is really, really, really important, because you might very well find a new discovery associated with a particular nucleus, which is really, really exciting stuff. So let me show you essentially what they do. This is what the process entails, step by step. They take single layers, slice cells into layers, and they scan it. Then that digital image is stored. Then the nucleus is manually traced by hand. Then all of those traced images are compiled together to give us that 3D image of a nucleus. So let's look a little bit about the tech behind that and see how it goes. All right, so what we're looking at, aren't they using 3D printing? No. There's no 3D printing in this. They're literally taking a cell. Now the cell that we're looking at here is what we call a HeLa cell. If you're familiar with the story of Henrietta Lacks, they did an entire film on that. It was based on the nonfiction book called The Immortal Life of Henrietta Lacks. HeLa cells were the first cells that they were human cells that they were able to actually grow in culture. And so I believe it's cervical cancer. And so these are cancer cells, HeLa cells. Now what they do is they take the cell, the individual cell, and they put it inside a little block. Then there's this diamond knife that slices off a teeny tiny section. And then they use the electron microscope to scan both in the X direction and the Y direction. Z is this way, all right? Z is going down. You got your X and your Y, right? And so they move down on the cell, slice off a layer, scan. Move down, slice off a layer, and scan. And so that gives us individual images of every single layer of a particular cell. Pretty nifty, huh? And it's all done one at a time, like the large-scale body. Yes. So that's what they do. And then they'll take all of the individual images of that circle that's traced and stick them together, right? That's what the whole idea is. So here is an electron microscope. Oh, the human body, sorry. Yes, that's fine. It's kind of like that, just one slice at a time, you know. Although that's a bit Hannibal, isn't it? With the one slice at a time if you watch the show. Anyway. So here is an electron microscope. This research could help with making specific drugs and vaccines better. Possibly. That could be an option for that. I would think the first role with this type of research is to develop a database involving all of the different shapes of nuclei associated with diseases. And then that could lead to better detection methods, because if you're able to see what shape is this nucleus, oh, it's this shape, it could be this disease. Vaccines are more associated with the cellular receptors on the outside of a cell. And so if you have a particular antigen, which is a protein expressed on a cell, that's what triggers the immune system. This would be really, really good for detection methods. So that's an electron microscope. It's pretty fancy, and they've got that cutting knife in there associated where they can slice it right off and scan it right away. Now, if you can imagine, these really little thin layers can generate thousands of images for one particular cell on any given day. So that's a lot of data, isn't it? That's a real problem too that they're still working on. So what kind of image do we end up with? Here is an electron microscope image for that particular HeLa cell that I was talking about. Notice the flashing green. That green is where the nucleus is. So here's the thing. If you go through in a day, slice up a cell, take all of these images, you're talking on any given day, you could have thousands of images. Mel's my favorite scientist. You're so sweet. Thank you. So on any given day, you could have thousands of images, and you have these scientists, tracing it out, could look for deformities in the nucleus and mitochondria. That's true. And yeah, that is a huge nucleus, isn't it? Well, this particular one is a cancer cell. And so scientists, they're spending all this time trying to draw out these particular lines, because guess what? Computer software and brain, computer software is not effective enough to be able to determine, is this a nucleus, or is that other circle there a nucleus, or is that other circle over there a nucleus? So it requires humans to go through and draw all that. But guess what? You don't even have to have a degree to do this. So if you just love science and, you don't even have a bachelor's degree in science, that's all right. Anybody can be a scientist in this case. This is their citizen scientist project, and you can go through and draw this. So that's how you can help. So here we have an image of an individual from the Francis Crick Institute who's sitting there literally drawing out every single circle. Now, you don't have to have a PhD in order to do this. All you have to do is just spend a minute or two. Yeah, organelle size varies with cell type and cell health. That's true. So you can draw it out on your own, and then this individual can be like, yay, I have people that can draw images for me. I can go and run these reactions for this new cancer test I'm developing. So this is how you can help. You can help her and other scientists that could be working on things, that are a bit more taxing, and kind of help relieve a lot of that load. And this is great for classrooms too. So if you're a science teacher, this is something your kids can do in your class, high schoolers, whatever. Or if you're working on your science degree, this is a good way to be able to differentiate organelles. Good study thing to do, right? So it's all right, and you can contribute and help in this manner. So then there we go. We got our flashing cell right there with the green all around it, and they compile all of these images together, and this is what we end up with. So this is an actual image that was generated from drawing around all of those circles, from all of those images. That's probably what, 1,000 lines, you think? That's a lot. And so they're able to do this 3D image and be able to determine this is what a nucleus looks like for this particular disease. And then this can go into a database, and then they can collectively over time store all of this information, and it can help lead to better detection methods of identifying different types of diseases by nucleus shape. So this is an actual image that was generated from drawing different pictures. And they have another one that's just come out that's exclusively drawn by people, like citizen scientists. So they've got those images out already for that. All right. So I've been talking about this project. Tens of thousands of images are available. Software is not as good as humans. You guys are way more important than software, because software is kind of crap right now for this sort of thing. But people are awesome with this. They're able to differentiate which is a nucleus and which isn't. This is good for classrooms and science enthusiasts. It reduces that scientific load. And guess what? You guys, the work that you do can contribute to teaching AI how to differentiate between nucleus and not nucleus. So this type of information can be used to generate software to help do this. So not only are you contributing to biological science, but you're also contributing to computer science. Double whammy, right? So that's pretty cool. It's exciting stuff. And then you might be like, hold on, what if I mess up? What if I trace the wrong thing? What if I trace the wrong thing and it's like not the right thing? Or, what if I get nervous? You know what? The software takes the average of what everybody assumes is the nucleus. And so it gives a statistical analysis. It is a lot of drawing. But if you have like 10,000 people that do like one picture a day, that's like 2 of those nucleus things already done. And they already have 3,700 volunteers. Yeah, so I mean, if you have like 10,000, and anybody in the world can do this. You don't have to live in London. You don't have to be in the UK. You can be anywhere in the world to participate in this project. So if you're afraid that you're going to mess up, fear not. You're not going to mess up. It's going to be okay, because the software takes the average. It gives a statistical analysis, and all the outliers will be visible. It gives you that nice little bell-shaped curve. So these are the messer-uppers here, and this is what the average is going to be. And they're like, that's pretty, this is what everybody thinks the nucleus is. And that's what they're going to go with. So don't worry about messing up. It's going to be okay. So you can be a big part of this citizen scientist project. How many cells have been mapped? My understanding is that they've already gotten a nucleus now. This is still a pretty new project, so you can jump in on the early parts of this. But so far, they've only had just a handful of nuclei actually put into that 3D imaging, because this is brand new. This is brand new science. And so they really need your help in order to pump out all of this analysis. And you're actually analyzing data. You're analyzing the data. You're saying, this is the nucleus, that's not. This is the nucleus, that's not. So your input is inherently valuable, because they scan all of this stuff and it's just sitting there waiting to be analyzed. And you can contribute to that project, that particular project, to help generate all of those images and kind of push this science forward. So it's a very important, it's an important scientific endeavor that's early on, and you can help contribute to that. So I'm going to show you where you can go to hop aboard this awesome train of cellular science. So you can go to Etchacell, twitter.com/Etchacell, or Zooniverse, Zooniverse, Zooniverse Etchacell. So I almost said Etchascale. That's not correct. It's Etchacell. So you can go to these particular websites and check it out. If you have any questions, you can always hop on Twitter, and I can link you to those websites. And if you're not following Etchacell, you can hop aboard. They update how many volunteers they have. In addition to, they show you the images that you contribute to, to help. So you can see what's going on and what the updates are associated with that. They were just at Manchester University showing their Etchacell project there. So I will say, all of this information is from the Francis Crick Institute, and I'm sharing it with you in an effort to try to help their research, because what they do is very important, and you can be a citizen scientist. You can be a real live scientist helping them out, analyzing this data, and moving forward not only computer software stuff, but also the biological sciences with stuff that's never been done before. And this is your opportunity to be a contributor to that. So that's pretty amazing. So I'm going to show you where you can find me. You can find me pretty much anywhere on social media, including the internet. Patreon.com/scientistmill. If you like what I do here, all of my content is free. Anybody can use it. Oh, you're welcome. I'm so glad you guys like this project. This is a very cool project, and I'm all about trying to boost scientific literacy, but also help scientists in the field. So if you like what I do, All of my content is free for anybody to use. I have a podcast now called Hey Scientist Mel. You can find me on Periscope, Scientist Mel, youtube.com/Scientist Mel, and I'm also on Facebook. So I have to take a moment and thank my patrons on Patreon: Tony, Zachary, Lauren, Anonymous, Carrie, Keith, Mark, Jen, Patrick, Carl, Tony, Anthony, Tristan, Paula, and Jennifer. Thank you so much for contributing to my Patreon account. You're extremely important to me. I'm able to have my website and generate resources for any science enthusiasts, including teachers. And I'm here for you to answer your questions and help boost this literacy. So that has been Etch Citizen Scientist Project. Check it out. I'll have a poll about what the next topic will be for next Saturday. I'll post it on my Twitter account, and you'll be able to vote about which topic I talk about, because I'm here for you. I'm here to help you learn and show you how you can contribute to science. So if you have any questions, hit me up on twitter.com. Send me a question on scientistmel.com. And happy sciencing. Thanks for hopping by. Bye.